so that now it will start coding for a stop codon. So to alter one codon, you can alter one base change, or you can insert 3 bases. Any of this, however you get to it. If you get to a position where normally you have a particular sequence and it gets converted into a stop codon, that's called a nonsense mutation. So imagine this is the normal sequence of a protein, of an RNA, UGC, UUU, and it codes for this protein, phenylalanine, phenylalanine, cysteine, phenylalanine. You may have a single nucleotide change, right? This is a missense mutation that changes the C to an A. When you change that C to an A, you get phenylalanine, phenylalanine, and stop. That's a nonsense mutation because you've inserted a premature stop codon. And now the protein will basically stop at that point, and the rest of the protein will not be made. And that's a very dramatic change in the structure of the protein. You'll have truncated the protein, made a much smaller protein, and very likely that protein is going to be malfunctioning. Okay. So nonsense mutations insert a stop codon, which result in premature translation termination, not transcription termination, translation termination, and because of that, you get a truncated polypeptide, which results in the loss of function of that protein. Okay, so important thing to keep in mind, there are no tRNAs in wild-type cells that have anticodons that recognize stop codons. So what happens if there is a mutation in a tRNA gene? So let's look at this, right? Here is a sequence of RNA, 5' to 3', and you get tRNAs base pairing. You have AUG 5' to 3', you have a CAU anticodon that base pairs with this, and this tRNA has methionine along with it. You have UAC, and that's read 5' to 3' by the anticodon GUA, and it will base pair with this and bring a tryptophan. You have an AAA, 5' to 3' on the messenger RNA, it's read in the anticodon 5' to 3' UUU, it brings along a lysine. And then you have a UAG, and that UAG is a stop codon, and there is no tRNA that recognizes that stop codon, correct? So translation termination is mediated by a specific protein, not by a transfer RNA gene, right? Okay, so what happens if you mutate this transfer RNA? Transfer RNAs are made from genes as well. They are RNA molecules that are made from a gene, just like messenger RNAs are made from genes. The only difference is the transfer RNA is not changed into a protein. It's not translated, but it's still made from a gene. So what happens if you make a mutation in a transfer RNA gene? What are the consequences? Especially a mutation in this region that is the anticodon region. So these are called nonsense suppressor mutations. Okay. It's a class of transfer RNA mutations that alter the anticodon sequence in transcription. And you can alter them in all sorts of weird ways. We're going to focus on one class called the nonsense suppressor mutations. So if you make a mutation in the anticodon sequence of a transfer RNA so that it now can interact with a stop codon, what happens then? So here is the DNA sequence. Imagine this blue box is the gene coding for a particular RNA. So normally, this blue region will be transcribed, made into an RNA. This RNA will fold into your cloverleaf transfer RNA. It will then attach an amino acid to it, and you get your normal transfer RNA, correct? But imagine there is a point mutation that happens right in the anticodon loop, right there. One of these nucleotides has changed in the anticodon loop of the transfer RNA. Okay. So imagine you have a sequence of a messenger RNA that goes UAC, UAG, UAA. So UAC is base paired with a transfer RNA that goes GUA, which brings tryptophan. Okay? UAG is a stop codon. There's no transfer RNA that binds it, but there is a protein that binds and stops more translation from taking place. And so tryptophan would be the last amino acid in a protein that's going like this. Correct? Okay, but now imagine that's your normal tRNA for the tryptophan that goes GUA. The messenger RNA is still the same, UAC, UAG, UAA. Here is your tryptophan RNA interacting the way it normally was, right here. But now you have a mutant tRNA. This mutant tRNA has acquired the ability to interact with this codon, UAG. It has a mutation such that instead of a G, it's been converted to a C. When you get these mutations in the tRNA gene, the tRNA now starts to pair with a stop codon. And when that happens, you basically are able to, you don't stop translation, you just keep making the protein. And so you get a longer protein. So this class of mutants in transfer RNAs that allow transfer RNAs to pair with stop codons are called nonsense suppressor mutations. The stop codons are called nonsense mutations, or nonsense codons. So a normal stop codon is called a nonsense codon. And when you make them suppress that nonsense codon, you basically are a nonsense suppressor. When you say suppress it, it means you basically don't recognize it. You don't read it correctly. You basically bypass it. So it's telling you to stop, but you have the ability to not stop and you keep reading through it. So these are mutations in transfer RNA genes. These are not mutations in messenger RNA or the protein coding gene, but mutations in the transfer RNA gene. But they will affect the protein, because the transfer RNA will bind a stop codon. Normally the protein would stop here, but because of this mutant transfer RNA, you will get a protein that will go beyond it.
Would that be just for that specific gene, or would they not be able to stop at any of the regular
Any of the genes that use this stop codon will be read through. So approximately 1/3 of all the genes would now be longer proteins, basically. Because there are 3 stop codons, and this transfer RNA will read one of those 3 codons inappropriately, and it will come through. Now, the effect is not as dramatic, because transfer RNAs, there are almost every transfer RNA, there are multiple copies. So there are usually, in yeast, for example, there's between 8 and 10 copies of every transfer RNA. One out of the step will be mutated. So you see how the ratio is reduced. So instead of 1/3 of the genes being read all the way through, it's probably more like 3% or 10% of the genes being read through. And the cell usually is sick as a dog almost. It takes much longer for the cell to grow when you have this transfer RNA mutation. But it does manage it, because you have enough wild type proteins being made that the longer proteins that are made get degraded or not functional completely. And usually when you have additions added at the end of a protein, it doesn't affect the function of a protein very much. It's just you're using a lot of energy because you're making longer proteins that you don't need. And so the cells can survive. And so these mutant transfer RNA is actually very useful in genetic tricks when you want to play games with specific genes. And so they become useful. So we use them a lot now as ways to increase production of certain segments of the gene. All right. So I'm going to play games with you with genetics and transfer RNAs and nonsense suppressors. So imagine you have a gene that looks like this: phenylalanine, and a stop. So let's imagine you first have a mutation in the transfer RNA that allows it to pair with this segment. So now, the tryptophan transfer RNA has a mutation. This allows it to pair with this stop codon. And so now, the sequence will go phenylalanine, phenylalanine, tryptophan, phenylalanine, phenylalanine, and then it reads a UAA, which is a stop codon, and it will stop. Correct? Okay. So now you get a gluten protein that is larger than the normal one that was synthesized. Let's take this example. Here is the sequence. UUU, UUU, CAG, UUU, UUU. It's making a protein that goes phenylalanine, phenylalanine, glutamine, phenylalanine, phenylalanine. If you now generate a nonsense mutation in this protein, so you're going to mutate this sequence here, CAG, and you're going to read UAG. Right? So here's a nonsense mutation. You get a truncated protein, correct? You take this mutant and you put in a second mutation in the transfer RNA. What happens if an individual carries both a nonsense mutation in the gene and a nonsense suppressor mutation in the transfer RNA? You basically end up with wild type in a certain amount of the type. So you are able to pair, even though you've generated a stop codon in this protein, because you have a suppressor transfer RNA, you're able to make the protein. It's made in less amount, so you've reduced the levels, but you still make it. And that's very useful. So we usually play these tricks with genes that are essential. So imagine you have a gene. This gene is essential for growth. If I were to put a stop codon here, the cell dies because it's not making the normal protein. But if I were to do that in the presence of a suppressor tRNA, then the cell survives. Because the tRNA allows a certain amount of this mutant protein to be read through, and you get growth. And so you can play these particular shapes. So you can read through, even though there is a stop codon. All right, so